My fellow biology students, in the following presentation, you'll most likely be learning about the immune system, but more specifically, the specific immune system. Hopefully, this will include macrophages, B lymphocytes, and T lymphocytes. I hope you enjoy. And introducing a very special guest, Professor Richard Head.
Hello there, class. How are you all today?
Good.
Yeah.
Woo!
Yeah. Oh, good. I'm going to take you through a process known as phagocytosis. It is one of the immune responses you'll get when a pathogen invades your body. This here is a phagocyte. More specifically, it's a macrophage. What do you think of that?
I think it's very interesting, but I'd like to know more, really.
Okay, I'll take you over to the first step of the immune response.
Excellent.
This little baby here, he's a bacteria. The phagocyte in this is undergoing a process known as phagocytosis. Here you can see lysosomes, which are bacterial digesting enzymes.
Woo!
That's pretty interesting, but I'd like to know more about the cycle.
Right. In the second stage, the bacteria have been ingested fully, and the lysosomes have binded with the vesicles that contain the bacteria. They will continue to destroy the bacteria and digest it very slowly. If it had been, it would probably be painful.
Poor thing. That's all well and good. What happens at the end, though?
At the end, the membrane of the bacteria is partly absorbed by the membrane of the phagocyte. This means it now becomes an antigen-presenting cell. It will go around your body looking up for other phagocytes and lymphocytes, which you will learn about later, and it will present these antigens, as you see here, by the big spikes, which are unusual compared to the original phagocytes, and it will basically let your body know that something is coming and it's not right.
Fascinating stuff. Thank you very much, Dick. Afternoon. I am Professor Phagocyte, and today I will be talking to you about lymphocytes. Right. All lymphocytes start out with here in the bone marrow as stem cells. Now, they can take two paths. They can either become B cells or T cells. Let's start with B cells. Now, B cells, they mature here in the bone marrow, hence the name B cells, to relate to the bone marrow. But anyway, when they mature, they become specific to produce a specific antibody. So, as a pathogen is caused by a macrophage, as you will see, it becomes an APC by absorbing the membrane. B cells, which fit the antigens, respond by dividing many times.
Okay.
And as they divide, huge number of identical B cells are produced. Some of these become plasma cells, which secrete the antibodies, but others become memory cells, so we have immunological memory, and they last for years. So, when they produce an antibody, these are proteins which the antigens bind to. And they, much like enzymes, antibody is specific to an antigen. So only one kind of antigen can actually bind to it and therefore destroy it. And now we're going to talk about T cells. Now, T cells, they actually develop, or mature, if you will, in the thymus gland. Now, when in the thymus gland, T cells develop things called T cell receptors. Now, these are specific to one particular antigen, so when they actually encounter it, they team up with the B cells to destroy the antigen, as it were. They activate when they encounter the antigen, obviously, in a host cell, for example, an APC. Now, the T cells come in two varieties: T helper cells, which encourage B cells to multiply, and T killer cells, which actively search for cells which have the antigen. Now, when they find these cells, they secrete these things called cytokines, which encourage phagocytosis in surrounding macrophages. And they also bind on and secrete toxins such as H2O2, otherwise known as hydrogen peroxide. The following demonstration has been deemed unsuitable for biology students, and therefore has been given the rating A for ridiculous.
This is
Pathogen. Macrophage. Yeah. B cell. T cell. The pathogen is hiding. pathogen has entered the body through either an open wound or bodily fluids. And as you can see, he's causing mischief. Unfortunately for the pathogen, the macrophage will not stand for this. And as you can see, the macrophage will try and deal with him. Eventually, he will engulf the pathogen. At this point, an APC is created. The APC now has receptors which B and T cells can bind to. This B cell has just found this APC. Unfortunately for this B cell, he is the wrong B cell. Boo! But what do we have here? A second B cell. And it looks like he's the right fit. Despite having the correct B cell, nothing can happen because our little B cell friend also needs a friend. This is where the T cell comes in. The partnership between the B and the T cell allows them to destroy the pathogen. Thus neutralizing the threat. Okay, wrong.
That's right.
This has been a public service announcement from Labtec 6. We hope you've enjoyed the presentation and certainly taken something from the experience. Thank you. And on a legal note, we would like to apologise for any inconsistencies that may have occurred during the presentation. We also cannot be held responsible for any inaccuracies that may have been presented. Should you have any problems, please refer to Dr. K, not us. This has been a public service announcement by Labtec 6. We hope you've enjoyed it and that you've learned something. Thank you.
Well, what is that?
Oh.